Alright, hi, it's Miss Nicholas. In this video I'm going to do a demonstration of the blood typing exercise that you'll be doing in lab with your instructor. Before we begin, I'd like to make sure that you're familiar with all of the pieces and parts that you're going to need to be able to do this. To begin with, you're going to need a tray or a microscope slide. If you have a slide, your instructor will tell you how to mark it and how to use it. Most of us will be using these trays that have 3 wells. The wells are marked A, B, and RH. That indicates what we will be putting into those wells. You're also going to need 3 bottles of sera. You have an anti-A sera or serum, which contains A antibodies. You have an anti-B serum, which contains B antibodies, and an anti-RH serum, which contains RH antibodies. Those antibodies correspond with the wells on the tray. These letters on the tray indicate which of the sera you will put in. You're also going to need an alcohol prep in order to sterilize your finger. You'll need 3 toothpicks. You're going to need something sharp, such as the lancet, to poke a hole in your finger, and if you bleed a lot, a Band-Aid. Alright, so now that you know what you're going to need to do this, I'm actually going to do it for you. Now, as many times as I have done this, it still takes a few minutes because it's not easy to make yourself bleed and poke something sharp in your finger. So just bear with me. Not many of your instructors can say that they've bled for you, right? Alright, so I suggest that you sterilize the side of your finger. There's a couple of reasons for that. One is the pad of your fingers have a lot more nerve receptors because that's where you touch things. That's what you use to manipulate things in your environment. Also, because you use the pads of your fingers more, they're also going to be more difficult to puncture. The skin is quite a bit thicker. So sterilize the side of your finger, put your alcohol prep to the side, and take your lancet. If you have this type of plastic lancet, you simply twist the top off, and then you have your sharp. Now, bear with me. It's going to take me a couple of tries. I told you it's not easy to do this. And you want to poke your finger. You start getting a little bit of blood. You may want to milk it a bit and put the blood into each of the wells. Now, it's very important that you put the blood in first so that you don't transfer your antibodies from one well to another. Now, I typically clot pretty quickly, so I'm not going to put the Band-Aid on. I'm just going to wipe the blood off and continue with our demonstration. All right, all's well. Now, I've done this many times, but I always check my labels before I put these into the wells. So, anti-A goes into the A well. I'm going to put a couple of drops in there so that you can see it well. You may not need that much when you actually do the demonstration. Checking again, anti-B goes into the B well. And being careful not to touch your serum or your serum droppers to the blood that's in the well. You don't want to contaminate it. The anti-RH into the RH well. Now, using those toothpicks, take the end of the toothpick and you want to stir the contents of that well. Put that toothpick to the side so that you don't use it again. Take a clean toothpick and stir the B, making sure you mix well. Discard it and take your 3rd toothpick and mix the RH well. Now, at this point, I'm going to put something light colored underneath it so that you can see the reaction better. And I will rock this tray back and forth a bit just to make sure that it mixes well. After a few seconds, you're going to be looking for the reaction known as agglutination. Now, this is not blood clotting. Blood clotting involves clotting factors. We are looking for agglutination, which happens when antibodies bind to the antigens on the cell surfaces. So you're going to see a clumping together of the cells. Now, you can see very clearly in the well marked A, all of the erythrocytes are bound together because we have bound those antigens and antibodies together. So there we see a positive agglutination reaction. In the well B, you can see that the blood is simply diluted. You do not have agglutination. So that tells you that there are no B antigens on those cells. We do have A antigens on the cells because we have agglutination. Now, the RH typically takes a little bit longer for the reaction to occur, and when you start to see the reaction, the agglutination is much less noticeable. So I'm going to rock this gently for a few seconds because I know that I should have agglutination in that well because I know my blood type. And sometimes if you apply just a little bit of heat to it, it speeds the reaction as well. So let's give it just a second and see what happens. You know, it's sort of like kids. When you want it to perform, it doesn't. And you can see a bit of agglutination starting to occur as the blood is running down the wells. And as this sits, you will see that the agglutination becomes more and more obvious. And after several minutes of sitting, you'll see that the RH and the A both have agglutinated. So this should tell you from your previous knowledge that if you have agglutination, that means that the same type of antibodies and antigens are present in that well. So because we have agglutination, we know there are A antibodies. We do not have, A antigens. Because we do not have agglutination in the B well, we know there are no B antigens because we put B antibodies in there in our serum. And now, let's see if we can see it a bit better. You still see just a little bit of agglutination in the RH, so we know that there are RH antigens in there that the RH antibodies bound to. So this blood type is A positive. Wherever you see agglutination, you know that that type of antigens are present. If you have questions, contact your instructor or one of us will help you understand it. See you next time.